Welcome to Chapter 9, The Fundamentals of Genetics. Today we'll be going over Gregor Mendel and his experiments and how to use Punnett squares. So let's begin with Gregor Mendel. Mendel was an Austrian monk-turned-scientist who focused on the studies of statistics and heredity, or the transmission of traits from parents to their offspring. His first experiment out of several involved the use of pea plants. Each plant had 7 distinct characteristics: height, tall, short, flower position, axial terminal, pod color, green-yellow, pod appearance, inflated constricted, seed texture, smooth wrinkled, seed color, green-yellow, and flower color, white-purple. Each characteristic was made of 2 traits. To conduct his experiment, Mendel collected seeds, recorded data on each plant's traits, and then planted the seeds a year later. He observed that on occasion white plants grew from the purple plant seeds, and short plants grew from tall plant seeds. This served as the base inquiry of his research. The process of self-pollination, the transfer of pollen from male anthers to female stigma on the same plant, as well as cross-pollination between separate plants, was used to answer Mendel's questions. Mendel manually cross-pollinated for selected traits in true breeding plants that always produced offspring with that trait. The parents were called the P generation, their offspring were called the F1 generation, and when the F1 generation self-pollinated, their offspring were the F2 generation. Mendel discovered that one P trait would not appear in the F1 generation, but reappeared in the F2 generation in a 3-to-1 ratio. From this, he gathered that traits are controlled by dominant and recessive factors. So, how does this happen? Well, this occurs due to the law of segregation, where factor pairs, also known as alleles, experience separation during the formation of gametes, and the law of independent assortment, which states that factors of a trait separate independently. Now that we know about Gregor Mendel's experiments and why traits are hereditary, we can look at genetic crosses and Punnett squares. These are used to predict what the offspring of parent organisms will carry in their traits. Genetic crosses have 2 types: heterozygous, where the alleles are the same and the organism is true breeding for that trait, and homozygous, where the alleles are different and the dominant one is expressed. The Punnett squares discussed in this chapter are monohybrid and dihybrid crosses. Let's discuss monohybrid crosses. In a monohybrid cross, a pair of contrasting traits is involved. For example, the 2 alleles for flower color in pea plants are purple, represented by a capital P as its dominant, and white, represented by a lowercase p because it's recessive. These letters are called genotypes. Here we can cross a homozygous white flowering plant with a heterozygous purple flowering plant. The resulting cross produces a phenotypic ratio with 2 purple flowering plants and 2 white flowering plants. The phenotype is the outward appearance. If you do not know the genotype of an organism, you can perform a test cross where the organism is crossed with a homozygous recessive. Sometimes alleles are not dominant or recessive and incomplete dominance occurs, where 2 traits are blended. The most common example of this is when 4-o'clocks breed red and white to produce pink flowers. Another type of cross is a dihybrid. In a dihybrid cross, 2 traits are compared. This can be applied to parrots and feather color with wing type. Say the 4 alleles are white and yellow for feathers, and spotted and solid wing types. Yellow and spots are dominant. Let's cross a heterozygous parrot for both traits with a homozygous recessive parrot for both traits. As you can see, we get 4 parrots heterozygous for both traits, 4 parrots heterozygous for base color and homozygous recessive for wing type, and 8 parrots homozygous recessive for both traits. The resulting phenotypes are yellow with spots, yellow with solid color, and white with solid color. And that brings this video to a resounding close. I hope you learned a lot about Chapter 9 and the fundamentals of genetics, and I wish you all good luck on your biology.